Good morning, my dear students. Let's start our new academic session. We will start from lesson number 1 of Science 2. So children, let's get started. Standard 10th, Science and Technology, Part 2. Lesson number 1, Heredity and Evolution. Let's begin, children. First, let us start with the introduction. Children, can you recall and answer me some questions? Which components of the cellular nucleus of living organism carries hereditary characters? Can you answer this? Yes, DNA carries hereditary characters of living organisms. What do we call to the process of transfer of physical and mental characters from parents to the progeny? Progeny means your children. The process of transfer of physical and mental characters from parents to the progeny is called inheritance or heredity. Which are the components of the DNA molecule? Yes, the components of the DNA molecules are two helical strands consisting of deoxyribose sugar, phosphoric acid, and a pair of nitrogenous bases. Now children, let's learn heredity and hereditary changes. You know that heredity is the transfer of biological characters from one generation to another via genes. Johann Gregor Mendel is the pioneer of the modern genetics. He discovered it. Around the period of 1886, it took a long time for him to understand the conclusion of his research about heredity. In 1901, the reason behind the sudden changes were understood. Due to the mutational theory of Hugo de Vries. Meanwhile, in 1902, Walter Sutton observed the paired chromosomes in the cells of grasshopper. Until then, it was not known to anyone. Research started in the direction of finding the nature of genetic material, when it was proved that genes are carried via chromosomes, through which 1944 a trio of scientists, Oswald Avery, Maclean McCarthy, and Colin MacLeod, proved that except viruses, all living organisms have DNA as genetic material. In 1961, the French geneticist Francois Jacobs and Jacques Monod proposed a model for a process of protein synthesis with the help of DNA in the bacterial cells. It helped to uncover the genetic codes hidden in DNA. Thereby, the technique of recombinant DNA technology emerged, which was vast scope in the field of genetic engineering. The science of heredity is useful for diagnosis, treatment, and prevention of hereditary disorders, production of hybrid varieties of animals and plants, and in industrial processes in which microbes are used. Understood, children? Now let's learn about transcription, translation, and translocation. Now, children, can you tell me? We need to sketch and explain the structure of DNA and various types of RNA. Let's start with DNA. DNA molecule is a double helix consisting of two strands. Each strand of this helix is made up of nucleotides. Each nucleotide is made up of phosphoric acid, deoxyribose sugar, and nitrogenous bases. Now these nitrogenous bases are of two types: purines and pyrimidines. The purines are of two types: adenine and guanine, and pyrimidines are of two types: cytosine and thymine. Adenine will always pair with thymine with double hydrogen bonds, while cytosine will always pair with guanine with triple hydrogen bonds. The helices remain bound due to these hydrogen bonds. Understood, children? Now let's start with RNA. RNA is the nucleic acid having single strand of ribonucleotides. Each ribonucleotide is made up of ribose sugar, phosphate molecules, and nitrogenous base. Adenine, guanine, cytosine, and uracil are the nitrogenous base substances which are present in RNA. Adenine and guanine are two purines, while cytosine and uracil are two pyrimidines. RNA is found both in nucleus as well as in cytoplasm. Now, according to the functions of RNA, it is of three types. First one is messenger RNA or mRNA. It carries the information for protein synthesis from genes on DNA chains in nucleus to the ribosomes in cytoplasm. Therefore, it is called as messenger RNA. Ribosomal RNA. It is the component of the ribosomes and it helps in protein synthesis. It is also known as rRNA. Then transfer RNA. It is present in the cytoplasm. According to the message of RNA, it carries a specific amino acid up to the ribosome as per the messages coded on mRNA. Therefore, it is known as transfer RNA, tRNA. Now, let's understand genetic disorders. They are caused due to abnormality in chromosomes and mutation in genes. Now, the reason for genetic disorders are increase or decrease in the number of chromosomes causing numerical changes, deletion of any part of chromosome, translocation of chromosomes, sudden change or mutation occurring in the normal gene, turning it into a defective gene. Mutations in more than one gene at the same time causes polygenic disorders. For example, disorders due to numerical changes in chromosome like Down syndrome, Turner syndrome, Klinefelter's syndrome. Monogenic disorders caused due to mutation. Examples are Tay-Sachs disease, galactosemia, phenylketonuria, sickle cell anemia, cystic fibrosis, albinism, hemophilia, night blindness are some of the examples of polygenic disorders. Cleft palate after repair, constricted stomach, spina bifida. It is a defect of spinal cord, as you can see here, are some of the polygenic disorders. Diabetes, blood pressure, asthma, obesity, heart disorders are also some of the examples of polygenic disorders. Now, with the help of RNA, the genes present in the form of DNA participates in the functioning of cell and thereby control the structure and functioning of the body. Okay? Now, information about protein synthesis is stored in DNA. And synthesis of appropriate protein as per the requirement is necessary for body. Now, these proteins are synthesized by DNA through the RNA. This is called as central dogma. mRNA is produced as per the sequence of nucleotides on DNA. Only one of the two strands of DNA is used in this process. The sequence of nucleotides in mRNA being produced is always complementary to the strand, that is DNA strand, used for synthesis. Besides, there is uracil in RNA instead of thymine of DNA. This process of RNA synthesis is called as transcription. Did you get it, children? The mRNA formed in nucleus comes in cytoplasm. It brings in the coded message from DNA. The message contains the codons for amino acids. The code for each amino acid consists of 3 nucleotides. It is called as triplet codon. Dr. Hargobind Khurana a scientist of Indian origin, has made an important contribution in discovery of triplet codons for 20 amino acids. For this work, he has won the Nobel Prize in 1968 along with two other scientists. Each mRNA is made up of thousands of triplet codons. As per the message on mRNA, amino acids are supplied by the tRNA. For this purpose, tRNA has anticodons having complementary sequence to the codon on mRNA. This is called as translation. The amino acids brought in by tRNA are bonded together by peptide bonds with the help of rRNA. During this process, the ribosomes keeps on moving from one end of mRNA to the other end by the distance of one triplet codon. This is called as translocation. Such many chains come together to form complex proteins. These proteins controls various functions in the body of living organisms and their appearance too. Living organism can produce new individuals like themselves due to genes only, and some of those genes are transmitted to the next generation without any changes. Due to this, some of the characters of parents are transmitted to their offsprings. However, sometimes sudden changes occur in those genes. Sometimes any nucleotide of the gene changes its position that causes a minor change, which is nothing but the mutation. Sometimes mutation may be minor, but some may be considerable. Example, mutation may cause the genetic disorders like sickle cell anemia. This is everlasting process and it is one of the proof for Darwin's theory of natural selection. Understood, children? Let's go to the summary what we have learned today. DNA. It is found in the cells of all living organisms except viruses. DNA contains all the genetic information that makes you who you are, and every individual organism has unique DNA like fingerprints. Understood, children? What is DNA? DNA or deoxyribonucleic acid. It is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. And where it is located? Most DNA is located in the cell nucleus where it is called nuclear DNA. But a small amount of DNA can also be found in mitochondria, where it is called as mitochondrial DNA or mtDNA. Now, what is RNA? RNA is a nucleic acid called ribonucleic acid. Its function is transfer genetic information from the nucleus, that is DNA, to the cell, that is proteins, for use. It makes up the ribosomes and it also helps assemble protein. Are you getting me, children? Good. Now the types of RNA. There are 3 types of RNA, each with a different job. Messenger RNA, that is mRNA, transfer RNA, that is tRNA, ribosomal RNA, that is rRNA. Each type of RNA has a different structure that is related to its function. Now mRNA, messenger RNA, is the RNA that carries information from DNA to the ribosome sites of protein synthesis in the cell. tRNA. tRNA or transfer RNA is a small RNA chain of about 74 to 93 nucleotide that carries amino acid to the location of protein synthesis. rRNA. It is a component of ribosome, the protein synthetic factories in the cell. Transcription. Cellular process in which RNA is synthesized, that is, it is produced using DNA as a template. It is known as transcription. Understood, children? Good. Translation. It occurs in the cytoplasm. mRNA goes to the ribosome. 3 bases on mRNA is a codon. Each codon codes for 1 amino acid. Anticodon on tRNA base pairs with codon on mRNA. In translation, tRNA then brings amino acids in proper order to make the proteins on the ribosomes. Now let's see what is mutations. Mutations are the changes in the sequence of bases in DNA or RNA. They are essential for evolution. To occur, mutations are the ultimate source of all new genetic material within a species. Most have no effect at all. Some are beneficial and some are harmful. I hope, children, you all have understood today's topic. Right? So, children, with this, we have finished with today's session. Please read the lesson after watching the video. Okay, children? We'll see you soon in the next lecture. Thank you, students.